Today I'm going to briefly go over how naltrexone, which is an opioid antagonist, affects the pancreatic carcinogenesis through the gut microbiome. So, data from our lab has recently shown that the microbiota promotes carcinogenesis in a variety of models. In this slide, you can see that when we give antibiotics, oral antibiotics to mice, tumor burden decreases drastically, not only in a pancreatic cancer liver metastasis model, but also in a melanoma model and also in a colorectal cancer model. Liver receives many microbial ligands from the portal tract. We also saw that the effect was not only limited to the liver, but also subcutaneous sites like primary cancer in melanoma-bearing mice as well as in pancreatic cancer mice. And we also found that this effect was replicated in orthotopic sites. That is, antibiotics significantly decrease cancer burden in orthotopic pancreatic cancer-bearing mice. With this in background, I would just like to touch upon the opioid crisis that is going on in the United States. Opioids are important analgesics in cancer as well as non-cancer settings. Their use is complicated by tolerance and dependence. Looming opioid crisis exists because of the abuse of prescription drugs as well as the availability of cheap heroin and synthetic opioids. And in this figure that we adapted from the New York Times, as you can see, many US counties are represented, and the ones in red correlate with increased number of deaths due to overdose of opioids. We work in Miami, and our hospital is right in the center of a looming opioid crisis. The first responders routinely carry naloxone and naltrexone because many people there die from overdose. We also, a few years back in gut, our lab showed that morphine, which is an opioid, promotes pancreatitis significantly. As you can see in cerulean-induced pancreatitis, a drastic increase in pancreatitis becomes remarkably worse with morphine. And our collaborators from the same institute showed that morphine also drastically changes the gut microbiome. So synthesizing all our previous 3 studies from the last 2 and 3 years, we wondered whether an opioid antagonist, naltrexone, can modulate pancreatic carcinogenesis directly or not. So we started by doing a very simple in vitro study where we gave variable doses of naltrexone in Petri dish to KPC pancreatic cancer. And we saw that only very supraphysiological doses of naltrexone could kill pancreatic cancer cells, most probably by lysis. As you can see, in normal physiological doses, the viability of pancreatic cancer doesn't change. However, when we gave very low physiological doses of naltrexone to mice, we found out that naltrexone almost halved the tumor cancer burden. This phenomenon was not only limited to subcutaneous pancreatic cancer, but also to liver metastasis bearing pancreatic cancer as well as in colorectal cancer. So we then asked, how does naltrexone modulate the intratumoral immunity? For that, we took the tumors treated with saline and naltrexone and passed them through a flow cytometry pipeline. And we found out that, surprisingly, naltrexone decreases the number of T cells that are coming inside the tumor. Now, T cells could be Tregs, they could be Th1, which kill cancer, Tregs, which promote cancer, Th2, which also promote cancer, and Th17, which also promote cancer. So we further dissected the identity of the cells and found out that naltrexone significantly decreases the Th17 cell compartment. Not only that, even the IL-10 secreting Tregs were also decreased remarkably by naltrexone. Both these T cells have been known to increase pancreatic carcinogenesis in literature. When we checked for the innate immunity, we found out that M2 macrophages, which also promote cancer, were decreased by naltrexone. So as a proof of principle, we repeated our experiments in 2 different strains of mice. We found out that RAG1 knockout mice, which don't have functional B and T cells, and we repeated the experiments in TLR2 knockout mice, which have a dysfunctional immune microbial sensor called TLR2. The effect of naltrexone tumorigenesis was abrogated. This suggested that naltrexone definitely required functional immunity. But it also suggested that the effect of naltrexone could possibly be mediated by microbiome because TLR2 senses microbial ligands. Therefore, we wanted to check whether naltrexone changes the microbiome or not, because this hasn't been studied previously. So we did a simple a pilot study where we gave mice fitsidextrin, which checks for gut permeability. And we found out that naltrexone decreases the gut permeability of mice, which also could mean that less number of microbial ligands are transferring from the gut to the systemic circulation. We then collected the stools from mice given saline and naltrexone and passed them through a 16S sequencing pipeline. And we found out that the alpha diversity, that is a number of unique taxa in naltrexone-given mice, were decreasing. And even in beta diversity, so on the right side of your slide, you see that each dot represents a different microbial community in one mouse. The green represents naltrexone, the red represents saline. The microbial communities are clustering separately. You can draw a circle, and they won't bisect each other, and which suggests that there's a significant shift in the microbiota. So we tried to check what species or what taxa are changing. At the species level, we found out that especially in naltrexone-given mice, two very beneficial bacteria, Akkermansia mucinifila, as well as Bifidobacterium, were increasing dramatically. Now, these bacteria are considered probiotic or good bacteria, and anything that increases probiotic is considered to be a prebiotic. So in our experiments, and we have reproduced our findings in lab again and again, naltrexone acts like a bona fide prebiotic. We also checked the species in a more granular fashion using a statistical technique called LEFSA, and we found out that naltrexone not only increased certain bacteria but also decreased certain bacteria, which many gram-positive bacteria, which we think are bad bacteria. So to establish causation, we gave mice either no antibiotics or oral antibiotics, which sterilize the gut of mice. And we found out that in control mice, that is without antibiotics, naltrexone worked. But when we gave those same mice antibiotics, naltrexone failed to work, which suggested that it required the microbiota. Now, antibiotics have many other, even the oral antibiotics could have many systemic effects too. And so we tried using germ-free mice, which are the gold standard experiment for any microbiome study. So we took a bunch of germ-free mice, took them out of their bubble, gave them the stool samples of normal mice and conventionalized them. And we gave them saline or naltrexone. And we found out that when we gave germ-free mice naltrexone, it failed to change tumor growth. But when we gave conventionalized naltrexone, tumor burden almost halved. This suggested again that the microbiota was an important intermediary between naltrexone and tumor growth. So in summary, we show that naltrexone modulates pancreatic cancer burden in a mouse model. We show that naltrexone also modulates intratumoral immunity, and its effect on cancer depends on functional immunity. And the effect of naltrexone modulates the gut microbiota, and the effect of naltrexone on cancer disappears in the absence of gut microbiota. With this, I'd like to acknowledge the important contribution of NIH, Minne Amrita, and Department of Defense, who fund us, my two mentors, the whole lab team, as well as, and above all, the animals with which I'm privileged to work with. Thank you.
All right. Thank you very much. Thank you for saving us 2 minutes. So we have time for a couple of questions. Anybody?
Fortunato Heidelberg. Did you measure also the microbiome in the tissue?
Yes. So in our previous studies, we have looked in pancreatic tumors, and we found out that pancreatic tumor may not be sterile, and that it harbors a lot of bugs, bacteria, which resemble the fauna of the gut. So we think that a translocation happens from the gut to the tumor sites.
Affected by the drug?
Yes, it does.
Okay.
Thank you.
All right. Thank you very much. I want to thank all the speakers for their outstanding job, covering topics from surgical management to neoadjuvant therapy to immunotherapy. I want to thank all of them for their outstanding talk. I also want to thank our co-moderator, Dr. Peak, and thanks for coming. That will conclude the section. Thank you.